Hello everyone, this tutorial is about building a phylogenetic tree using any phylogenetic tree building software. So before going into or jumping into the phylogenetic tree building, first we should know that why we are doing this and just revise the small concepts we have already discussed. And after the end of this tutorial, I will send you an exercise via email, and you have to perform following this tutorial. So why we build a phylogenetic tree? Actually, it depends on the question we have in our mind. For instance, if we want to see a relationship between species, if we want to see a relationship between genes, gene families, that how those gene families are evolved, then we use any phylogenetic software and get the sequences of that gene family which we want to look for, and then build a phylogenetic tree. But there are a few steps, as we have discussed in our previous lectures, that first we should have a set of sequences, and these sequences can be of any population, any gene family, any species, that depends, any disease patients, right? So we have a set of sequences, and then we have to do multiple sequence alignment of these sequences, and based on these multiple sequence alignment, we build a phylogenetic tree, right? Now just we should be quick to go to the next step. For instance, now my question is to see that for example, my question is in this tutorial is how a protein family let's select a protein family, serine gene, right? Serine incorporated protein 1. Let's select it. So how a protein family serine is evolved from invertebrates to vertebrates. Now this is our question. How the serine family is evolved from invertebrates to invertebrates. And now if you have to answer this question, you can clearly see that we should have the details about this family, right? This family. Then we should have the protein sequences of this family, and then we should have the list of invertebrates and invertebrates in which we want to look for the evolution of this protein family, right? So step by step we move towards towards our answers. So first step is to go and find the sequences which are present for serine families in Ensemble Genome Browser. Mainly I select Ensemble Genome Browser because it's very non-redundant information and very accurate, as we have discussed in our previous discussions, that it has manually curated and automatically curated all the reference genome sequences currently available. So I think I should not go into the details about how to extract the protein sequences. If you want to go and to know how to extract the protein sequences, we have already shared this tutorial with the title Blast and Ensemble Genome Browser Tutorial for Beginners, and the topic is extraction, gene sequence, and finding orthologs, right? Now you know what is an ortholog. Ortholog is a gene which has been evolved through speciation event, right? So here we are talking about protein sequences, and if you go through the presentation, we selected different species, and any gene sequence we search. For example, here this is an example of TP53. Okay, but when you are going to do the protein sequence alignment, right? Because here we want to identify the protein sequences of serine family in vertebrates and invertebrates. So here you have to select in this example, you have to select the protein sequence and insert it here, and then select protein, and then at the search against option, you can select the number of whatever number you want to select for the blast, and just click all those species and then click OK. And here at the blast, the search tool which you have to select, here it is selected T blast N. Here you don't have to select T blast N, you have to select, instead of T blast N, you have to select blast P, because we are doing protein alignment, right? So I don't think I should do this again, because it's already available on my channel, or you can see on my page. So yes, so for that we have to have a protein family sequences, and then how it is evolved from invertebrates to vertebrates. And then one more thing is that after having so there is a possibility, I have already downloaded for your ease the list of sequences of this serine family. So here you can see that I have organized it in a very user-friendly way. For instance, here you can see that this is the Homo sapiens. For example, Homo sapiens, Homo sapiens are humans. And then there is a serine C1 gene information, and then there is the ensemble gene ID of that species. So this is the format, FASTA format, right? So there are 3 genes. After doing the multiple blast tool and finding the orthologs, we identified that there are 3 genes. One is serine 1. Where it is that? If I want to write it here. Serine 1, serine 2, and serine 3, right? So using these 3 genes, which are part of the serine family, I first used serine 1, and then did blast against all the species, which I will give you in the exercise, and then serine 2, and then serine 3, and then I combined all the FASTA format sequences of serine 1, 2, 3, and then generated the multiple sequence alignment. So now just jump into the exercise. For instance, now we have a multiple sequence alignment tool. This this tool I have already explained in a tutorial that how to download this tool and use it for the multiple sequence alignment. And then this is the example of all the species. Sorry, just skip it. Yeah, this one, right? So I just downloaded all the sequences using BLAST, basic local alignment of proteins. What is the one technical thing you have to do? That, for example, if you want to download the serine C1 and serine C2 and serine C3 sequences in multiple species, first of all, you have to download the sequence of serine C1, right? Just go and select serine C1 in the human box, just check the human box and then click serine C, and then go and then download the protein sequence. That is also explained in one of my tutorials. Here you can see that if you go into this analytics, here you can, so not analytics, in the BLAST you can find, in the ensemble playlist you can find the example of downloading the protein sequence, right? But maybe I think it's here in the beginning. Yeah, in the beginning you know how to download a sequence. And by downloading this first serine C1, then if you want to search it in macaca mulatta, for example, macaca mulatta is monkey, gorilla is gorilla, pantroglodytes is chimpanzees, muscle. If you want to download these sequences in these species, you have to do BLAST P, right? You should have a sequence serine C1 and then blast it in the BLAST P in the species I give to you. And then for serine serine C2, you have to download the sequence first, extract the sequence, and then do the blast with this all the species. And then for the serine C3. So this is just an example that how you have to go, and then you have to download the sequence, and then blast it in the BLAST P in the species I give to you. And then for serine C2, you have to download the sequence first, extract the sequence, and then do the blast with this all the species. And then for the serine C3, so this is just an example that how you have to go through the process of finding the orthologs. After finding the orthologs, you will save all those in a FASTA format in one file like this, and then open the Mega software, and from Mega software just go into the align and build an alignment, right? And here I have already done, so I will close it first so that you can have a bit of understanding. Yeah. Now here it is create an alignment. So I click OK, create an alignment. Then are you building a DNA or protein sequence alignment? We are building a protein sequence alignment. Then click the protein. Now this is the box where you have to insert all the sequences. So you go there, check or control all, control copy, control paste, right? Now all the sequences, there are 37 sequences which I have used for this example. And if they are in FASTA format, then it will easily be copied here, right? Now I want to do the multiple sequence alignment. Then I select all these sequences, control all, and then go to alignment. In alignment, there are different types of alignment, you know, cluster, W, muscle, T, coffee. But this is, you can just select any of them because we are not, we are just having a vertebrate and species. So, and these are the most widely used alignment software. So cluster W I selected, and from cluster first just go take it all the parameters as default, then click OK. After clicking OK, now you can see that it's first building a pairwise alignment, and from the pairwise alignment it will build a multiple sequence alignment. And this multiple sequence alignment, in this multiple sequence alignment, you know, we are aligning 37 different sequences. So now this is an alignment file of this serine family, and each color, if you just want to scroll it here, you can see each color represents different protein amino acid sequence, and there are some dashes between that which are known as gap. So we are just focusing on the, okay, now this is this is a sample serine, right? So serine, go into, I want to save it in some very easy, okay, example, serine family, save. Yep. Now it is saved. Now I want to build a tree. Now I want to go, I have to go to the file where I have saved it, and from the save file just go and click all files, and is it saved or not? Let me save it first here. It's saved, yeah. Okay, go right here. Yeah, so now all files, it's you just select serine. Now it will, yeah, it's now in the, run by the Mega, and I selected what? I have selected neighbor joining method. So bootstrapping, just we didn't have talk about bootstrap right now, so we just excluded no bootstrapping, neighbor joining method. And then there is substitution of amino acid method. There are different types of models as we have discussed to calculate the distance. You know about B distance, that's Hamming distance, and then there are other methods. We have discussed a couple of basic concepts. So as we have we discussed B distance, to just select the P distance, and then keeping all the options as it is. Now, yeah, this is the tree using those 37 sequences, right? So maybe it looks very difficult for you for the in first look, but it's very simple. If you go into the basic, here you can see the details. That, for example, this is the first part of the sequence. This is serine C1, Homo sapiens, then macaca mulata is monkey, then gorilla, pantroglodytes is chimpanzee. So here's a cluster of primates, and then there's an outgroup of Ecoscabulus is horse, Mus musculus is mouse, and then it is Loxodonta africana, right? So these are different mammals which are not primates. So it's there's a cluster of mammals outside that, and from the mammals then there is an outgroup of, you can see here, birds and frog. So it's for the tetrapods and birds. So there are three clusters. One is the primate, then there is a cluster of mammals, and then there's a cluster of birds and frog. So birds and frog and are also two different. It should be clustered separately, but we have given the few number of sequences. We have selected very few sequences. If we want to differentiate between the birds and the tetrapods like frog and lizard and these species, then we have to select more species here so that it may give us a more detailed differentiation. And then from this outgroup, you can see here there's an outgroup of fish. Tachyfugu. Fugu is a fish, and Gastrosteus aculeatus is a stickleback. So this is a very beautifully devised cluster, you can see. And then there is so this is a serine 1C1 cluster, and then there is an outgroup of another cluster that is of serine C3. So it's the serine C1 and C3 are clustered together. Again, you can see here that Homo sapiens and pan-troglodyte chimpanzees, and there is a cluster of primates, and then there is an outgroup of what? Mammals, and then from the mammals there is an outgroup of tetrapods and fish, and then there is a last outgroup of what? Last outgroup of fish. So here you can see that these two are clustered together at this time point, right? And then there's another, you can see here is outgroup of another third serine C2 here. You can see that serine C2 is again very beautifully defined, organized protein structure. Like Homo sapiens, gorillas, pan-troglodytes, macaque are clustered together. Then there's an outgroup of mouse. It's also a rodent or mammal. And then there's an outgroup of two other mammals, and then outgroup of bird, and then frog, and then there is a cluster of fishes. Here you can see that there are multiple two types of serine 2A and serine 2B, and then there is a stickleback 2A and stickleback 2B. So, and all this cluster has an outgroup of the invertebrates, Saona, intestinalis, and Drosophila, and the species. So you can see that all these species are having are evolved after vertebrate conversion from the invertebrates to the vertebrates. As we have discussed in our previous lectures and basic terminologies, the outgroup is the most distantly related group. So the outgroup of the whole tree is actually these three invertebrates, which suggests that in the invertebrates there was the most distantly serine was present. And you can see here that all the serine 1, 2, 3 are not present in Drosophila and Saona. It means that there is a event of called duplication after invertebrates. So what is a duplication and speciation event? If you see again from after dissecting, I think you have an idea that they are clustered together. That we have discussed the differences between speciation and duplication. Speciation is actually when the speciation event occurs, like the new species evolves, right? And what is duplication? Duplication is when a copy of when a gene is duplicated and it makes multiple copies within a same species, right? So if the gene is evolved through speciation, it means that the gene has been traveled from one species to another through the speciation event when the species were evolving, new species were originating. And the gene is duplicated means that within a species there are two copies of a gene, right? So now here you can see that in this cluster all these points where you can see, if I can highlight this, oh no, okay, all these events, when the gene is not changing but the species is changing at every node. You know what is a node, what is an internal node, what is an external node. We have discussed in the phylogenetics terminologies. You can go and just go through the basic lecture there. So these are actually all the speciation events, right? And what is a duplication event? The duplication event is the point where the new gene is, the copy of a same gene is, originated or duplicated. For example, where you can find this in this tree. Here you can see that serine 1, this whole cluster before this point till is for this one species, that one gene, that is serine 1. So all these are, this is one gene. So other duplication at this point, you can see that at this specific node there is another cluster of serine 3 gene. So we can say that all these are the speciation time point, because there is no difference of a gene but difference of a species. But at this point there is one duplication, duplicated serine 1 and serine 2 together, right? Then there is another duplication after this cluster, that is the presence of serine 2a at this time point, right? So after this you can see that this whole third cluster of the serine C2 is present, and all these genes are of the serine 2. So if this is confusing for you, I will make a very simplified figure. You can clearly go to here. You can see that if you see this one, that in this they have explained that there is one serine C1. It was the first one, right? This is serine C1, and it was then it is duplicated into, if I just make a gene tree, and then it is duplicated into, not this is the time points I will explain, not this first one, this is the second one, but these are the 3 species genes which are actually evolved. So this is the most distantly, means it's the most ancient. So this is the time point at which this was the first duplication, and this is the second time point for the second duplication to make these 3 genes. But where is this time point in the tree? I'll explain to make it more clear to you. This is the time point here. Yeah, this is a time point where the serine 2a make another ancestor of. So at this time point, this is which is highlighted in red, there is a gene that is called serine 2a, which was duplicated into the hypothetical ancestor of serine C1 and serine C3, right? And then there is another duplication of at this time point which divided this ancestor into serine 1 and serine, at this point serine 1 into serine and serine 3, right? So now you know that there are 2 duplications actually, duplication 1 and 2, which originated or you evolved this family into 3 different genes, and these are the overall gene. But there are some species specific duplications. For instance, here you can see at this time point in the tree, if I make it again, here you can see that Takifugu rubrics of serine 2a and 2b, right here. This is a speciation event between serine 2a and stickleback of serine 2a. But this time point here, you can see that there is a fish specific, family specific duplication, and the serine 2a is divided into 2a and 2b, and stickleback gastrus aculatus serine 2a is divided into a and b, and this is fish specific duplication, right? So we can say that there are overall there are 2 duplications to divide this into 2 duplications for in overall gene families, 2 duplications, and one fish specific duplication, right? So there are multiple duplication events which avoid this family, and then there are a number of speciation event, right? Speciation events. Number of speciation events, right? Speciation events. How will you write it? Speciation event. Speciation events, right? So if I explain what I mean by number of speciation event, what I mean is, actually you can see that there are number of speciation events, 1, 2, 3, 4, 5, 6, 7. All these are actually the speciation events, because the species are changing but the gene is not changing, right? So I think now it's very clear that how you use how to download I started from the very basic, that how to extract the sequence. You can go to the first tutorial to summarize things up for your understanding, right? Extract, first point is, right, if we go to extract gene sequence in first of all humans, right? First of all, then blast P for in this exercise to find orthologs, right? And then after finding all the orthologs, depending on the number of orthologs you have, then you have to do multiple sequence alignment, right? And from multiple sequence alignment, generate tree, and from that tree in this exercise you will find the orthologs, paralogs, right? I think now it's very clear to all of you. I will give you the exercise through email, and you just have to perform that. But first you have to see the blast, how to extract the gene sequence information, protein sequence information, and then finding the orthologs, and then doing the multiple sequence alignment and generating a phylogenetic tree, right? So we have just took an example for neighbor joining method. You can select any other maximum likelihood method as well, and you can interpret the whole protein family. Thank you very much. I think it's getting longer, so hope you guys we have a very detailed discussion about how to generate a tree. Let's see how you come up with your exercise and how you perform this. If you have any questions, you can send me via email, or you can post a question outside beneath this tutorial, and we can have a very healthy discussion. We can have also a very healthy discussion here. Thank you very much. Hope all of you are fine, safe, and happy. Bye bye.